Welcome to MOOC NPTEL course on Interactomics. Determination of interaction kinetics is one of the most striking application for SPR biosensors, and understanding of kinetics is essential to evaluate the strength of a biomolecular complex interaction. The label free real time detection allows kinetic analysis to be performed by monitoring the interaction as a function of time over a range of analyte concentrations, and then fitting the whole data set to a mathematical model describing the interaction presenting the kinetic parameters. For example, association and dissociation rate constants. Affinity constants, which exhibit the strength of binding between the molecules, can be derived either from the rate constants or from steady state binding analysis. A kinetic analysis experiment can be set up in a Biochemical experiment in two ways: multi-cycle kinetics, where each analyte concentration is run as a separate cycle, regenerating the surface after each sample injection, and single cycle kinetics, which runs a series of analyte concentration in one cycle with no regeneration between sample injections. Today we are going to perform the kinetic analysis for the protein beta 2 microglobulin with anti-beta 2 microglobulin antibody using multi-cycle kinetics. So, let us have the lab experimental sessions now.
We will start with low immobilization protocol set up for anti-beta 2 microglobulin, as a lower immobilization level will be beneficial in this kinetic interaction analysis.
We have to redo our immobilization. In this case, now we are selecting flow channel 3 and selecting flow channel 4. Flow channel 3 will be a blank immobilization, and flow channel 4 we will immobilize anti-beta 2 microglobulin at 1200 RU for a kinetic experiment. Here again we will highlight prime before run and keep the sample compartment temperature at 25 degrees centigrade, and we will go to the next tab. Now we will add vials with the rack positions and the volumes.
As per the volumes listed in the table, we will prepare the reagents for immobilization in a similar manner as demonstrated earlier in lecture 33. We prepared a working concentration of 30 microgram per ml of beta 2 microglobulin antibody from a stock concentration of 1 mg per ml. Using an immobilization buffer of 10 millimolar sodium acetate pH 5. We prepared HIPIS EP Plus as the running buffer, which was connected to the system followed by priming. EDC and NHS were prepared to be used in 1:1 ratio for surface activation. 1 molar ethanol amine HCl pH 8.5 was prepared for blocking the free ester groups on the surface. Lastly, 50 millimolar NaOH was prepared, which will be used to remove the electrostatically bound ligand. We prepared the above mentioned solutions, transfer them to appropriate sample vials, place them in the suitable rack and start the immobilization run as we did in lecture 33. To save time, we shall now proceed directly to analyze the data obtained from immobilization of beta-2 microglobulin antibody.
So this is a blank sensogram. After immobilization, we will see the reference surface is blank immobilized. Here you see the activation of the chip by EDC NHS and blocking of the surface with ethanolamine. So this will be the blank immobilization. Now we will go to another surface or another flow channel. Going to the next flow channel, FC4, highlight it. This is a sensogram for active surface immobilization involving protein. To start with, there is a pre-concentration estimation of the required amount of the ligand, wash with the sodium hydroxide, activation of the surface with EDC and NHS mixture, pulsing of the protein for the required number of RUs, and then block of the extra sites with ethanolamine. The immobilization level is actually determined from this level subtracted to this level. Now this will conclude our immobilization procedure. We have successfully immobilized the required level of ligand for our next experiment. We will now set up a wizard for kinetic analysis. To start with, we have selected from the wizard kinetic template, selected the flow path 4-3, chip type CM5, sample, and the flow is to regeneration. Once we are set up with this template, we say next and startup cycles are selected at 5 with HPS EP plus buffer. The sample contact time of 180 seconds is selected. Flow rate of 30 microliter per minute is selected. Dissociation time of 600 seconds is selected. Coming to the regeneration solution is glycine HCl pH 2.5 with a contact time of 30 seconds, flow rate of 30 microliter per minute, stabilization time of 0 seconds. And we now go on to the next tab and fill in the name of the sample as beta-2 microglobulin with a molecular weight of 11,800 and concentration with 0, 2, 4, 8, 16, 32 and the repeat of 8 nanomolar again. We say next and prime before run is highlighted. Now we have the rack positions this way. We have regeneration buffer here, startup here with the samples here.
In today's kinetic assay experimental setup, we will be using HIPIS EP Plus as the running buffer. We will be preparing a total of 5 concentrations of beta microglobulin starting from 32 nanomolar, 16 nanomolar, 8 nanomolar, 4 nanomolar and 2 nanomolar using 2-fold serial dilution in the running buffer and 8 nanomolar concentration in duplicate. We will be using HIPIS as zero concentration negative control for the reaction as well as for the initial startup cycles. For regeneration solution, we will be using 10 millimolar glycine pH 2.5. We shall now proceed to set up our experiment for kinetic analysis. We will now work on the reagents required for the kinetic interaction analysis between anti-beta-2 microglobulin and beta-2 microglobulin. Once again, we will be using HIPIS EP Plus as the running buffer, which will also be used for the initial startup cycle. We will be using our protein stock concentration of 100 microgram per ml to prepare 5 different concentrations of protein, including 32 nanomolar, 16 nanomolar, 8 nanomolar, 4 nanomolar and 2 nanomolar. We will be preparing this concentration series in 2-fold dilution using the running buffer starting from 32 nanomolar solution, which means we will add 300 microliter of 32 nanomolar concentration with 300 microliter of buffer to prepare 16 nanomolar concentration. We similarly continue with the dilution series for rest of the concentration. One of the concentrations, that is 8 nanomolar, will be run in duplicate, and we will also include a 0 nanomolar concentration in the experiment. For regeneration, we will be using glycine HCl pH 2.5 as the regeneration solution. All these samples and reagents have been transferred in the required volumes in these specialized tubes and will now be inserted in the appropriate rack and then into the system for the kinetic interaction analysis.
So from the template here, we have prepared the sample rack with the different vials. To start with, we have startup placed at E1 position, and we have the different concentrations of beta to M with the concentration mark from positions at C1, B1 to B6, So 32, 16, 8, 4, 2 nanomolar, and again 0. Once we have placed them, we will also have glycine 10 millimolar pH 2.5 regeneration solution. Put at the A1 position. Now we close the rack. We go on the screen, eject rack, say OK. We say next on the tab. So it takes 3 hours 34 minutes with 200 ml of minimum buffer, and say start. You can see here it says running kinetics affinity. Takes 3 hours and 34 minutes with a priming of 6 minutes. Once the immobilization is done and the experiment is being run, this is how the data will end here, and from the data we can go to tools, Biocore T200 evaluation software, so this is how the data is generated. You see all the startup runs here, and we go on overlay to select sample data. Now we go on tools, sensogram adjustment, report point, baseline, again to the tools, color by concentration, now you can see all the data is differently colored. You see the rate of association, dissociation and regeneration with the legend of the different concentrations of beta to M injected. Here as well, like binding, we will right click on the mouse and cut away this portion. And now this is our actual data with the different concentrations of beta to M injected on the surface. To evaluate this data, we can go into the kinetic and affinity mode. Select surface bound. The data is shown here. We can go to the, here you can see the different cycle numbers, different concentrations of the beta to M. At a flow rate injected was 30 microliter per minute, contact time of 120 seconds and dissociation time of 300 seconds. We have selected 2-1 surface. The ligand that is immobilized is anti-beta-2 microglobulin and the sample is beta-2 microglobulin or analyte. Interaction performed at 25 degrees centigrade. Now we go on to this and see next. If at all there are unwanted spikes, can be removed in this region or in this tab by simply highlighting that area and right click and highlight that unwanted region and say remove selection. Similarly, we could take away these spikes by highlighting that area with the right click of the mouse and say remove selection. Now that there are no spikes, we can proceed to fit this data in the kinetic mode as you can see on the tab. Now you see here 1-to-1 binding and parameters here, fit here. So we could place mouse on the fit and press start. So data is fit with the black lines superimposing on the colored lines and the following information is generated. If you see the quality control tab, report tab, the residuals tab and parameters, each of them has data from this experiment. To start with, we go onto the quality control tab and see here all the three are in green with kinetic constants are within instrument specifications, are uniquely determined and no bulk contributions found. We have learned from our theoretical sessions bulk contribution will be more if there is a mismatch between buffer and sample. Now we go to the next tab, report. You can see lot of information that is generated here. The on rate is given here. Similarly, there is an off rate and there is a affinity or a binding constant provided here. The R max is also given. The various concentrations that was used to set up the experiment are provided here. Flow rate for this experiment is provided here. The change in the refractive index due to differences in the sample and running buffer are provided here, and a quality control parameter like chi-square and U value are also shown. So Kd refers to the affinity of the two molecules, which is 2.6 into 10 to the power of minus 9 molar or 2.69 nanomolar affinity. Rmax or a maximum number of binding sites available at 24. Chi-square of 0.0901 should be below 1 at least. U value below 25 is always considered to be good. We go to the next tab, residuals. The residual should be aligned between the red lines, preferably between the green lines. And here you can see that the data is completely within green lines, and that indicates the superimposition of the black lines on the colored lines is very close. Now we go on to another tab, parameters, and find lot of information which are nothing but the standard error calculated for the data. With this, we conclude the interaction of anti-beta 2 M to beta 2 microglobulin at 2.6 nanomolar affinity.
We will start a new topic on protein, a small molecule interaction study in next lecture. Thank you.